All right, so now we're going to cover incomplete dominance. So, incomplete dominance occurs when either allele is not completely dominant over the other. So, there is no real dominant allele. It's like they're both kind of equal, so to speak. So, it occurs when the dominant allele does not phenotypically cover the other allele. So, the heterozygous phenotype, when both alleles are present, the phenotype is not one or the other, but actually a mixture of the two. So the best way I think I can explain this is use an example of red and white flowers. So for red, so red and white flowers are an example of incomplete dominance. And so if you have an allele that is big R, it'll be for red. And if you have an allele for big W, it'll be for white. So if you see a red flower, or let me do it this way. If it's homozygous for red, the flower would be red. If it's homozygous for white, the flower will be white, phenotypically white. But if you have the heterozygous, the red and the white, like one's not dominant over the other. And so what happens is you end up with pink. Why? Because the combination of red and white is pink. And so whenever I do these problems, I always write this out because this is super helpful for me. I always write the genotype with the phenotype. So let's do a problem. So it says cat fur is determined by incomplete dominance. So that means one is not dominant over the other. Cats can have black or tan fur. The heterozygous condition TB results in a cat with tan and black spots called a tabby cat. What would occur if, that should be an if a tan cat is crossed with a tabby cat? So I'm going to write out all the possibilities. So if a cat is black, that means it's going to have both, right? Black. It would be homozygous for black. If it was tan, then it's going to be homozygous for T. So notice I use the first letter of B and the first letter of T. The heterozygous, which is T and B put together, and really it should be BT because it should be alphabetical. But anyways, that's not a big deal. That we call that tabby. So when I do incomplete dominance, I always find it's helpful to write this out. So then it says, what would occur when a tan cat, which is big T, homozygous for T, crossed with a tabby cat, which is TB. So I'm just going to do that cross. So big T. So 50%, or 2 out of 4, right, will be the genotype homozygous for T, and that will be tan. And then 50% will be TB, so it'll be tabby. Okay. All right, so let's try another one. So you could go ahead and try this one on your own. So it says if two tabby cats are crossed with each other, what is the likelihood that they'll have a tabby kitten or a black kitten? So here's the question. We want to figure out what percentage will be tabby and what percentage will be black. So two tabby cats. So remember, if you remember, TT was tan, right? TB was tabby, and BB was black. So we have two tabby cats, so two of these guys. So TB, oops, let me make that bigger. Let me do my Punnett square first. Cross with another TB, so all right, so what's the likelihood to have a tabby kitten? The first question is, so 50% will be tabby, and then black kitten, 25% black. Okay. All right.